get all that?
We're in Syracuse.
If we could touch base.
Either way.
Good. Hi, I'm really glad to be here. I've heard a lot about this conference, and it's also really great the way it's set up. We have a whole lot of people who I've known for a while, and we are all in sort of different disciplines, which I don't know if it was planned, right? So Dr. Haffman, who sort of welcomed me back to the Finger Lakes where I was an undergrad when I first got back here, is a geologist who's worked here for a long time, done a lot of comparative work, and then Dr. Howard actually introduced me to nutrient ratios, which is a lot of what I've studied since then, nutrient ratios of plankton as well as exotic species, when I worked in the lab next to his. And now you've got and so he's a biogeochemist, now you've got a biologist, a biological limnologist, and then you get sort of an engineering DEC person at the end, so you'll see this lake from a whole bunch of different perspectives, and I think we're all actually linking together pretty well. Today, I want to talk a little bit about something that was supposed to be sort of a small project. I'm working with Upstate Freshwater to help them parameterize their model. I'm looking at some plankton, which I'm not going to talk that much about. The plankton that I've been counting is looking pretty similar to the plankton in the past, so it's not as interesting as what happened with the mussels. So as you guys have had these HAB problems, but so have a lot of people all around, not just the Finger Lakes, but also sort of the whole northern region of North America and of Northern Europe. There's a nice picture that one of our alums, Tim Snyder, took here. He's a local. And so I think there's increasing evidence. We thought right in the 70s, the people that I learned from thought they'd sort of fix the eutrophication problem by reducing phosphorus, as Dr. Howard sort of said. And now we're starting to see all these HABs appear. And so, you know, what's happening? I have given a talk to high school students and to some college students saying, like, this is not your father's or your grandfather's eutrophication, right? Something else is probably going on, because you guys, for example, have a lake that has pretty low phosphorus concentrations, and yet you're having these algal blooms. So maybe something else is going on in addition to phosphorus, which we know adding phosphorus can also cause problems. So we're seeing even in lakes like Skaneateles and Owasso Lake that have low phosphorus, as you guys have been hearing, some lakes having these cyanobacterial blooms. And so what else has changed, and why are we getting these blooms? I think it's a big unanswered question that probably is unanswered a bit because there are a lot of several different causes. And because I'm a biologist, and because I think part of my message here is going to be like, this started out as being something that I thought was going to be a simple thing to just do and help Upstate Freshwater Institute parameterize their water quality model. So the species matter, and also observing the habitat that things are in matter. And because we're terrestrial, pretty big terrestrial organisms, we live on shore, we boat in the lake, and not a lot of people spend time, say, where the mussels are at the bottom of the lake. Like, who would want to do that? Maybe only me. And the pond weeds, right? So first, let's say also the plankton that are causing these problems. So this is a biological graph that sort of shows all of life on Earth. There's sort of three big domains: the bacteria, another kind of bacteria that's sort of not distantly related, and then all of the sort of complex celled organisms. And the further away you are from some other line on here, the more different you are biologically, the more different origin you have. So all of the trees, like if you eat plants at all of those things are in this one branch of land plants. And then all animals, like a horse or my dog, are in this branch, right? And you wouldn't confuse, I've tried to think of how to explain to my students, like, how different phytoplankton are when things like they're all just these tiny single-celled things that, you know, float around, they look the same, and why can't we just fix it? So you wouldn't think that, like, my dog would behave like the tree, I hope, and you wouldn't confuse them. Okay, if we look at where on this different branches of life things that we call phytoplankton are, it's like we have the cyanobacteria that are in this whole different group here, and then all of these different groups basically are things that are phytoplankton. You know, and so they're as different from each other as my dog is from the tree, okay? And that's part of why it can be really hard to control algal blooms, phytoplankton blooms, because all these things have real differences. So they matter in lake ecology. They matter when we're trying to manage things. And if we were tiny, tiny microscopic creatures, I mean, life wouldn't be as fun, but we would know that these different things were different. If you were walking along the street and that was person size, you would know that this was not the same as that, and was not the same as this or that, right? But because we just see that the lake looks a little green, or the lake looks blue-green, or the lake looks brown, you know, we don't realize that there's hundreds of different species of things there, and it may just be one or two things that are growing out of control or blooming that are causing the problem. If those things happen to be containing some toxins, like some of the cyanobacteria do, then it causes a real problem for human health, not just the aesthetics of your lake, right? So there's a lot of possible factors that we know can affect cyanobacteria. So sometimes I think people get in arguments, right? It's N, it's P, it's, you know, the problem is there's a lot of factors that can favor one group, like the cyanobacteria, over other kinds of phytoplankton. And not all cyanobacteria are going to make toxins or bloom. So there's a lot of possible causes of harm to algal blooms. We don't know them all. We don't always know when different ones are going to dominate, but that's what we're all trying to put together now. That's why it's complicated. The easy one that you reduce algal growth is if we reduce the phosphorus into the lake, we sort of know that, but there's other things that come into play to choose between those different players, those different kinds of phytoplankton. So there's crazy complicated things that you took them around the class, you know, about physical processes and all kinds of things. But you know, we know that the ice is coming off of the lakes earlier and coming onto the lakes later, and so we have a shorter ice on time. Like I wanted to go on the lakes to collect some quagga mussels for some students, and you know the ice is sort of sketchy right now here. If you went up to Oneida, it's not bad. You could get on a little bit, but the ice comes off. That means that we have a longer time for lakes to warm up. And we're seeing all around the northern region that even though the air temperature hasn't increased a lot, the surface water temperatures, when you start warming the water, like you guys know when you're swimming in the summer, it's warm, that lighter warm air floats on top of the cold water that I had to go on when I was diving. That's heavier and it's below it. When that's up at the surface for a longer period of time, because there's less time with ice, then that warms a little more. And we know some things that you have the same nutrient conditions, right? You have the same amounts of nitrogen and phosphorus, the same ratios of nitrogen and phosphorus. Cyanobacteria often win competition with other phytoplankton just because the surface water is a little warmer. And if you look at Dr. Halfman's records, right, we have some summers where we're routinely getting now over like 24, 25 degrees C temperatures. That's going to help cyanobacteria win, even if we manage the nutrients the same way we have before. So that makes things difficult, but that's, you know, we can still solve this problem. So we also, because we have warmer water, that means there's a bigger difference between the surface water and the bottom water temperature, and that means it's harder to mix that. And it means that some researchers, like Karsten at Cornell and others, have the people who were working on Skaneateles Lake with the buoys there also found that on Skaneateles Lake you have this warm water and cold water and the thermocline. The winds blow and they sort of make a wave internally that breaks on the shoreline, and that can resuspend nutrients from the sediment, and that can maybe fuel some of the blooms like in Honolulu Lake or in Skaneateles Lake or other lakes. And so what that does is basically all the years when we put so much phosphorus into the watershed, as well as nitrogen into the watershed, and built up these really organic, rich sediments and stirred those up, right, with the water breaking. That resuspends what I call here the legacy phosphorus. I didn't invent that term, but I think it's a good term. Like it's a legacy of 100 years of land management and not thinking that we needed sewage and septic systems and those kinds of things that caused algal blooms and caused erosion that came into the lake, grew algae, released sediments that all sank to the bottom, and that's this really nutrient-rich pool of phosphorus in the water. That's actually the biggest nutrient-rich pool of phosphorus in the lake is that sediment layer that's down there. And I think that's going to come into play when we think about these mussels as well, more than I would have thought. And like Dr. Howard was talking about, when we change the nitrogen and phosphorus ratio, that can also change the balance between cyanobacteria. and other algae. And one thing may be these invasive mussels that we have. We've had zebra mussels for a while, but we also have quagga mussels. And so not all of you are old enough to remember when quagga and zebra mussels came into the lake. I was an undergrad in the Finger Lakes region, and I actually dove for Dr. Johnson down at Cornell for one of my many jobs, helping him harvest invasive plants. And we were diving in Cayuga Lake. There were no zebra mussels then. I'm really old. There were no zebra mussels then in the lake. There were some zebra mussels in the Great Lakes, but they hadn't reached Cayuga Lake. These days, you know, you grow up and you know that there's going to be zebra mussels near shore of a lot of lakes, and then quagga mussels came in after that, more recently, and they haven't invaded as widely in the region yet. They're still in the middle of an invasion. So people who are old enough may remember that when those mussels came in, say up at Oneida Lake, it was really dramatic. The water clarity really increased. You could see that Secchi disc, you know, more than a meter deeper than you had before, because those mussels are like little phytoplankton vacuuming machines, right, sucking that up, and then they're basically excreting, they're putting their waste products into the near shore zone. They also have these shells that are irritating and wash up on shore and hurt your feet. But that sort of, the fact that the bottom of the lake, we call it the benthic zone, is sort of where they're living and also where they're excreting. They don't have a septic system either. They're releasing the nitrogen and phosphorus from the phytoplankton back in the water. And a lot of you may have seen, you know, these blooms of these filamentous algae like Cladophyra, and this also could perhaps help promote algal bloom. So the idea is you went from having lakes that maybe had too much phosphorus, like Oneida Lake, and were really green lakes in Lake Erie. And then for a while after the mussels came in, they got way more transparent. And a lot of people were excited about that. Like I never thought, I mean, I was diving in Cayuga Lake for my job, but I wasn't thinking like, oh, it would be a really good idea to go have recreation on, you know, the St. Lawrence River to see something, because you have to basically run into it. But now you can go up there and the water's clear, and you can see the wrecks there, because really the mussels, it helped that we had nutrient control, but you got an order of magnitude like more water clarity after the mussels came in, because they're also taking those plants, the phytoplankton and the photosynthesizers in the water, and they're sucking them out of the water and putting that nutrients on the bottom. And near shore, that grows a lot of pond weeds, as you would call them, macrophyte big plants, as I would call them, because that sounds so much more scientific. But we're growing all these benthic algae and all these pond weeds. Makes it hard to navigate through there. And they're reducing they're changing the balance of who's the winning primary producer from, you know, the things that are in the water to the things that are on the bottom. A lot of people have done a lot of work on zebra and quagga mussels about how they eat, and there's some contradictory results, but they definitely don't just eat everything indiscriminately. You know, a lot of them may select against some distasteful kinds of phytoplankton. Onondaga Lake, the open water after the mussels came in got clearer, and it also had lower phosphorus. All of that stuff was being released at the bottom, and if you went to the back covering when the zebra mussels were just settling on hard surfaces near shore, they came in first, and the zebra mussels predominantly will just settle on a hard substrate, rocky areas. They tend to be pretty near shore. When they did that, you would see the concentrations very near shore of phosphorus be, you know, say 3 to 10 times higher than they were offshore, because of the excretion, right? It was the near shore zone was the toilet of the zebra mussels, essentially. So we have this high phosphorus near shore due to mussel excretion, and that grew a lot of plants and grew a lot of Cladophyra. It could sometimes promote some near shore harmful algal blooms too. And there's always a lot of filamentous algae growth and more pond weeds. And also the studies really differ about what they're eating. Depends on the conditions. It also depends what nutrients they're releasing based on what kind of the nutrient content is. So they take what they need to make their own bodies, and they may release more or less phosphorus depending on what's in their diet. So one contribution to HABs is rescinded mussels, both by them not necessarily favoring eating them, so they don't take them in, or they release them as pseudofeces. But also, they may make these concentrated pockets of phosphorus at the bottom and for zebra mussels around the edge of the lake, where they're excreting and raising nutrient concentrations. And then if there are algae that can go to those concentrations and suck up those nutrients, they might have a competitive advantage over others. And even though you think of phytoplankton as just sort of being at the mercy of currents, it's the whole idea of plankton means wander. Plankton can't like swim against a current, but that doesn't mean it can't move. So some plankton are flagellated and they can go up and down. Cyanobacteria can regulate their buoyancy by regulating sort of gas content in their cells, basically making a little like flotation device to go up and releasing that flotation device to sink. They can't all do it, but a lot of them can do it. And so the mussels are releasing, they can alter whether nitrogen or phosphorus is dominant. And the cyanobacteria can often also move up and down, as Dr. Hackman and others mentioned. And so they may move down at night to photosynthesize, up in the day, down at night to take up nutrients, up in the day to photosynthesize. And I'll say, when people were saying like, oh, like the bloom was, as Dr. Hackman said, and someone else said, you know, the bloom wasn't there. Well, when we were out doing our dive surveys, it's not the only time. I've seen this in several lakes. You know, the bloom, it looks fine. We go out in the morning, we're like, yeah, we can dive now. We're not in a harmful algal bloom that's going to kill us, you know. And so we're out there trying to get our dive samples, and we go down to the lake, and it's sort of murky at the bottom, and you don't think about that too much because you're like, it's often murky at the bottom, but not always. And then as it gets lighter and lighter out, and we start seeing the murky layer move up, and the first time it happened, when it got close to the surface, I realized, okay, the harmful algal bloom is moving up in the water, and we should bail now because we're in like really high concentrations of a potentially harmful algal bloom. So I've seen it like myself. Sometimes I have to like think of filming that when I see it because it's pretty dramatic. So when you guys in say the late August and September, we're having some surface blooms in say the north end of the lake. It would be fine for us. We went out, you know, in the morning, and the lake would look good, and we'd start diving, and then we'd have to abort taking those samples and come back a different day because by midday you had blooms, or even late morning you had blooms that were coming to the surface that were worrisome. So these migrating cyanobacteria may have an advantage because they can get those nutrients in the sediment. So if the mussels are releasing those, they may be facilitating that there's a little layer of really concentrated nutrients in that area. So there was a survey done here by Jim Watkins and Lars Lundström and others at the Cornell Field Station, Don Dittman from the USGS and John Hackman, and they went out to the lake and they took ponar rods. And a ponar, if you haven't used it, is like a little construction device on a miniature scale that you set a trap here, open it up, and then when it hits the bottom, it scrapes the bottom. And I don't know if this will work, but these guys had a really good little movie, which I don't think but of how, so you can see, like they lowered it down. We're just going to show part of it, so you can see how it's going to be a little relevant. It's not going to work. Okay, so you lower it down, and it's, well, take my word for it. They're going hopefully it won't crash the whole time. So you lower it down, and what you can see it does is that the two halves basically come together and just scrape maybe the top, 1 or 2 centimeters of the sediment and grab all this stuff that's on it and bring it to the surface. And that's how most people, most sensible people, sample for mussels. Oh. Wait, or should we? That's like a lot of bad choices. That's what you get when you try to be all fancy and like user friendly. great game. Trust me, you can watch the movie if they have it, if it works. So, what this team did was they went along your lake and they surveyed along sort of the central axis from north to south at 10 sites, 10, 20, 30, 40, 50 meters, and then back up the other side. And they did that once in June and once in October in 2007. And some of you guys may have heard of this. Okay. And this is what they sort of found. If you look at their data, this is going from the north end and then down to 10 meter deeper station, so like about 30 feet deeper station, 30 meter, 40 meter, 50 meter in the middle of the lake, and then back up to 30 feet station was their sort of shortest site. So what they found was that there were zebra mussels, a lot of zebra mussels in the lake, right? So this is under a meter squared of surface area, so just a little bit bigger than a yardstick by a yardstick. You would find in, sort of a slightly shallower area, it's still pretty deep, you won't be walking there, right, like at 30 meters deep, 30 feet deep. You get about almost 3,000 of those meters in that small space, right? That's a lot of mussels. And then they found similar things in the fall, except that they found there were a little bit higher densities of them there. And they found that there was a really diverse native invertebrate population on the bottom of the lake, and that's really good for fish food. But what they found was that, and I will show those data here, where there were lots of mussels, they didn't find as many of those sort of nutrient-rich, easy-to-digest invertebrates living at the bottom of the lake that fish would eat. And they found those mostly in places where there were not a lot of mussels at the time. And so they expressed the hope that they hope that the zebra mussels the quagga mussels would not invade because they had been invading a lot of the other lakes. And they attributed a lot of the positive community of small animals in the lake bottom to not having quagga mussels. Because in other lakes, the Great Lakes, places that we've seen quagga mussels invade, some of those other really nutrient-rich invertebrates that are really high-quality fish food start to be reduced because the mussels are occupying all that space in the floor and altering sort of the oxygen and the chemistry. The quaggas look a lot like zebra mussels. They have some differences. The students who are helping me, whose names I'll put at the end, are counting all these guys and spending hours of their life doing this. They really know these differences well, but you can tell the difference not always by the color. Their quagga mussels also get a little bigger. They have slightly different shapes, but in terms of the lake, what matters is zebra mussels really live on soft surfaces primarily, on hard surfaces primarily, on rocky areas, you'll see if there's a rock in the middle of a soft area, they'll all be on the rock. Quaggas can live in soft substrates. They can go all the way down to the mucky bottom of the lake. They also can live in colder water, and they are really dense, and that dense amount of animals living at the bottom of the lake that are all doing what we're doing, right? Using up oxygen and breathing out carbon dioxide. That lack of oxygen actually chemically can make phosphorus more soluble in the water. And so that also means that the legacy phosphorus from the watershed and all the production in the past may be more likely to be released into the water because you have all those organisms that are respiring and potentially making phosphorus more likely to come out of the sediments and enter the lake again. And generally, quaggas also filter more of the phytoplankton than the zebra mussels and excrete a little bit more. But again, there's hundreds and hundreds of studies and they all differ a little in parameters. So my work was supposed to be helping Upstate Freshwater Institute to parameterize their model for the water quality model for your lake, which they did a model for Cayuga Lake, and this is a diagram that Dave Matthews sent me from that, where they looked at how zooplankton excretion when they're consuming phytoplankton and releasing those nutrients, and and muscle excretion, and then the consumption of zooplankton by mussels as well, small zooplankton, how that affected nutrient release, and that nutrients, as you've heard from earlier speakers, then might help promote blooms. So when they did this model in Cayuga Lake, they found that the mussels were, the quagga mussels and the descendants were increasing the phosphorus release. I believe something like doubling the amount of phosphorus. Maybe it was halving again, so can't get any specifics. So my job was really for my students and I to count phytoplankton and mussels and assess the mussels in the lake so they could make their model of your lake and decide how much does the excretion of the small animals in the water, the zooplankton, and these mussels at the bottom of the lake actually contribute to them like re-releasing phytoplankton nutrients back into the water. It sounded straightforward. And the other question was, like, are there quagga mussels in Owasco? People thought there probably were. Quagga mussels, the presence of quagga mussels coming into a lake has often been correlated with increases in the algal blooms, although it's not a perfect correlation, so people thought maybe they were partly to blame. So we decided to do a survey. So a lot of people, now that images are getting cheap, you know, you can buy GoPros or GoPro knockoffs pretty cheaply, have started doing aquatic imaging. So, you know, back in the day, this was pioneered in places that people would like to dive and that are generally clear, like marine systems, so coral biologists and people came up with photo methods to go along transects and take images and see how many animals and the kinds of animals were living at the bottom of the lake. And so that's been brought into freshwater systems more and more. It's harder, the optics are harder, people don't want to dive there necessarily. And, you know, if you can do that, and if you could potentially like image a bunch of the bottom of the lake, like Lou McCaffrey has a little rig that he can go around and image the bottom of the lake with that camera. We have a different one where we have the normal sampling you would do with a half-meter little square, a half-meter on each side. It's got a little GoPro rig on top of it, and little feet that allow it to settle down into the sediments and just get the surface image, but it's the same idea. You know how much of the substrate you're imaging, and then you can have a little program these days automatically count how many of them are there. ImageJ, funded by like NSF and NIH, our government dollars, made this free program you could download if you wanted to count, I don't know, like checkers or marbles. You could have an image and have it trained to do that. It's not hard to do at all. And then what we were supposed to do was not just take pictures, but see, like, will this really work? You know, because no one really tested as to, you know the Ponar data, you take pictures, you see the mussels. Can we have an algorithm that works really well then to just make images all around the lake, which is pretty easy to lower something tedious, but pretty easy to lower something down, take a picture, run it through a program once you have the method. So we were supposed to dive on about 20 to 30 quadrats, 20 to 30 of these little plots that we image and collect the mussels, collect nutrients first above the mussel bed, so that if I could run them and see, like, are there more is there more phosphorus, more various forms of phosphorus and nitrogen above the mussel beds that have more mussels in them than in other places. And then we would, after we collected the nutrients, and that's actually sort of fun. You can't use like a little bottle that is full of air and then open it up the way you would at the surface, right? Because that would either crush or it would pop to the surface because it's so buoyant. So you basically use a bag that's rinsed and washed, and then put that into the normal sampling bottle when you get to the surface. So we'd hover, another diver and myself, safety teams of two, would hover above the sediments and basically open the plastic container without touching anything, lower it, and then sample the mussels that were imaged. And so it was supposed to be pretty straightforward, but just like a lot of things at first, they always tell you, like, when you're in high school, the first step of science is observation, right? And so this is the kind of thing we all observe the quagga mussels pretty much by having the Ponar go down, like we couldn't observe in the movie, and bring us back what's there. And we don't very often like go down there and look at what's going on. I should have known like something would go wrong when we did that, but I cavalierly thought humans just understood the mussels, even though we never really went down there. So we would sit up in the boat, we had a sonar that had measured the depth, waypoint where we were going down. We had the GoPro rig with a line, take images of it. You can see the edge of the whole quadrant here. This is actually a real planty one, so that's not a good thing. We'll talk more about that. It's a 1/2 meter by 1/2 meter on the side. And then we would pull up everything on there. We had a tender on the boat. We had 2 really good dive lights illuminating the whole thing. And we'd have 2 life divers go down the line. One would go take the nutrients, and then after that was done, we could disturb the bottom and take up all the mussels. UFI got our bottles right away when we were done. And then we collect the mussels in this fun way of basically mucking about at the surface, pulling them in. We had gloves. We also, our dive master at ESF, our dive safety officer, we practiced all this in the pool, right? So we had tried like little colanders from the grocery store. It's fun being a field scientist because you go to Home Depot and other places and they're like, can we help you? And you're like, well, probably not. Like, I'm not using this for the purpose you intended. Like, what's sieve in the kitchen section would be great for like sitting up a lot of I don't even know. So we went down and once you start stirring up the bottom, you can't see a darn thing, right? So you also have to be not claustrophobic, and by feel, you take all the mussels off the surface. So we have these double mesh bags. They have to be mesh again, because if you had gas in it, then they would float to the surface, and that won't be what you want because you're still at the bottom collecting it. We collect every single mussel. You can feel that. It's actually better than collecting plants, which is the other thing we do sometimes. We'll talk more about that. And then we could bring that back, freeze it in the lab, and have some other poor students do the sorting that we'll talk about. So on the boat, they're in the cooler. They get labeled in their bags. They have their associated nutrients. We give the water off to UFI to run. We store the images. And then the mussels right away are frozen and then get counted through by these great ESF undergrad students and one grad student for how many total mussels are in the bag. But also, you have to decide if it was a mussel shell or was it live when it was collected. So was it hinged and tight, and you look, there's actually, flesh, there was a mussel in there, or is it something that was not intact at the time of collection? They also identify quagga versus zebra, which when you see the numbers of mussels that there are, you'll see that this is a really laborious process. And then we'll be taking a subset, either 50 or 100, we'll calculate what's needed, to see like the size of them. The zebra mussels can be really small, just a few millimeters in their first year, and then grow pretty large. So the filter rate's a bit dependent on body size to estimate that average size for the population. And so the places we went last year, North End, so we did a couple transects here. UFI helped do this. This is an area where you get a lot of algal blooms, and here as well off the point, and then the south, and then this really steep, and I don't know if people really realize, you might if you're a fisherman and you see your sonar, how steep it is there, but it's also really rocky. They have like a little bit of remnant zebra mussels there. So we did these transects at the end of the season, August and September, and we still have to actually do the deep ones that our dive safety officer, actually I'll give him a plug, Jason Meany, who runs Deep Stop Scuba Shop in Township 5. The reason that he couldn't do these this year is because he was opening his new scuba shop, but he's the ESF dive safety officer, really great scientific diver. So he's going to collect those really deep samples, because not everyone, it's not safe for students, and it takes special gases to do those samples that are really deep. So Upstate Freshwater Institute ran the nutrient samples immediately after collection, and we're actually close to a third done with the sample processing for mussels. It's been hours and hours and hours, weeks and weeks and weeks of undergrad sampling time. So I'll show you, like a transect of worth of data. But we also, just in that, have found some actually surprising things. And I will say that actually we found some things while we were out there that were surprising, and did a little bit of sort of side thing, few samples. So here's what we found. This is mussels in that meter squared. So think like yard by yard by yard, right? Okay, this is disturbing, right? So these are zebra mussels, and these are quagga mussels. And we're going from the shallow area. We started a little shallower than the survey that had been done 10-ish years ago. And the zebra mussels are still hanging out a bit near shore at about the densities they were. You'll see that in a second, which is still a lot. It just doesn't look like a lot anymore. 5,000 mussels in that meter square. And now the quagga mussels have come in. So you have quagga mussels, and you have a lot of quagga mussels. You may not see them near shore, but if you were to go 20, 30 feet deep and beyond, you would see a lot of them. So we go to, our deepest sample was about, that we did was almost 90 feet, and there were still a lot of quagga mussels there. So there's lots of quagga mussels. And they were peaking sort of around this 12-meter deep depth in this transect, and just visually that, and the numbers that they're getting in, that's also the case generally. If we compare that to that previous survey, here's just the zebra mussels when we had it, right? They had about 3,000, used to have about the maximum here of 5,000 of zebra mussels, and now you got tons and tons, so almost like 10 times more mussels in some places of the lake than you had even when you had all of those quagga mussels. Okay, but the thing we found out when we went down there, so we thought this was simple, we're taking these mussels off the top, because we thought they only really lived at the top, because we sampled at the Ponar and that's where they are, right? But no one's looked. So the thing that happened, I went down, the first site, I was not the nutrient collector, I was the person who collected the mussels. And so I go down, I'm collecting the mussels off the surface, and I feel that there's more mussels down there. So I collected these mussels off the surface, and I feel down, and there's more mussels, and more mussels. I could put my hand, my arm, in the sediments, all the way up, like, well, probably further, but that's as far as my arm went, okay? So it was sort of like a slurry, like if you make a slushy, or, you make some kind of, super supposedly healthy drink that looks nasty, right? But you can stick your hand in it and it's sort of semi-coagulated, right? It was not, a lot of the sediment was really what we call unconsolid, like really loose sediment that you could put your hand through. And there were mussels down there. And we're like, well, what the heck? And we're like, well, we're supposed to, for this contract, like collect mussels at the surface so we can calibrate these images. That's what we should do. We can't just collect mussels until the end of time. We don't even know if they're alive. So then I didn't say anything to the assistant. I said, like, pay attention at the next site, because we switch each time, and we have the tender at the top. And so Bob, who was the student who had just graduated, who was the other diver, at the next site, he's like, looks at me, and then we get this, he's like, there's mussels that are down there, and you can put your hand all the way in the center. Like, yep, so it wasn't just that. So that's how the deep part of the lake was in all the places that we were sampling, like super unconsolidated samples. So what we decided to do was go tiny bit rogue, and just for like 3 sites, do a little extra on our own, and take a little quadrat, little square that we had for a different thing we had been doing before, and was still in the container, a 1/4 meter on a side, and at 3 of those sites, take the sample we were supposed to take at the surface, like we were, and then take all the mussels, and we agreed at the point in our arm that we both could reach up to, that we would collect to there if we found them, and put those in a different bag, and then counted how many mussels are in there, and just check to see, like, are they afterward, that's what the students and us did, to see if they were still alive. So, we also, because we were going shallower, found mussels on plants. And that earlier survey, right, hadn't done anything that was shallower than 10 meters. And when we were taking anything in soft surfaces, we would get plants, and we sort of knew this from doing some other plant work. There were mussels on the plants. So mussels hang on the plants. The images also, I mean, I don't have to, we'll do it all, but like it's not going to work, because in the image you see like the plants all mushed down, maybe this much plants that are being mushed down by your quadrat, and you see some mussels at the top, but then when you pull the plants out and we're pulling the mussels off, there's like layers and layers of mussels that can't show up on the image because you're basically seeing plants. You just see green. They're very laborious to remove. The 4 students who are doing this deserve some kind of extra credit because you're pulling them off. And currently, we're counting them. They're pretty high numbers, like in hundreds at the minimum to thousands for some, again, per meter squared of plants. And so you have a lot of mussels, almost as much as just on the mud, that are on these plants. Some of them are really small because they're like the larval stages. These mussels have a larval stage that floats around the lake, how it disperses. But there also are some bigger ones there that recruit to the plants, and then they can actually move to another place when they're done. And so that started us thinking as well, you know, if anyone eats blue mussels, or you've seen aquaculture where people are harvesting blue mussels, it's not as extreme as this. But, you know, the plants in the nearshore zone as well may also be containing enough mussels, and a few people have quantified some of this in some places in the literature too, but not as much how much they're filtering on these plants, and acting like a little zebra and quagga mussel farm, because we've seen both of those on the plants, and maybe allowing the mussels to access some of the phytoplankton that are in different layers of the water, and that selective filtering and stuff is occurring there. We don't know, but the plants and the sediments had us really thinking that we were not totally understanding what was happening, right? Because the mussels are down in this deep sediment, and then they're alive. What are they? Did they move up and down? We don't know. They're alive. Like the ones that were there were alive. The sediment is unconsolidated. We know that oxygen can maybe increase phosphorus. If we're stirring up the sediments, we have that buried legacy phosphorus that might be important. We also think, if you think about this, we're in the nearshore zone. We also have mussels here. So there's whole populations of mussels that we didn't know existed and don't really know what happens to them in the lake. So when we count them, just in these 3 samples, so this was in that same site where we collected the surface mussels that averaged, pretty high, over 10,000 mussels that were in the surface area. And then we took that other one and we also equilibrated that to the whole thing. There were over 5,000 on average mussels, so over 6,000 mussels per square meter living, alive ones. We don't count the dead ones, so they sort live and dead ones. They sell zebra and quagga mussels living in the deep sediment. That's as many as there were zebra mussels before in the nearshore zone, live mussels in the deep sediments. I don't know what they're doing, but it seems like maybe that's important to figure out. Mostly it was quagga mussels that were in this. On average in these 3 samples, 14% was zebra mussels that were alive there. They were attached to other quagga mussels or shells of quagga mussels, but they were alive down there too. But there are lots of them, right? That's a lot of mussels, 5,000 per square meter that are alive. That doesn't count all the dead shells that we also quantified. So about 20% of the number of the subsurface mussels are subsurface. We don't have a big enough sample size to know. It's pretty variable. Is that constant enough that the image can still work? I don't know. We didn't know that was even in the scope of the study, right? So this legacy phosphorus that's in the sediments, how is it being affected by this potential for mussels, respiring, maybe releasing through excretion and releasing through abiotic processes where phosphorus is more soluble when there's lower oxygen? This is the importance of observation firsthand, I guess. Like, as much as it was a distressing day when we realized they were down there, it was also like, hey, what are they doing? Like, what did they eat? Did they come out? I don't know. So it's a whole thing we didn't even know was a habitat or something. So right now, we've got this team of great undergrads sorting these guys to zebra and quagga. We're cross-calibrating them and sizing them. And that'll go to UFI for the model. And testing if these images can be calibrated. We get all of that data set. We basically reserve some of the samples where we don't know, we don't know how many are, we know, but we don't add it to the model and see if the algorithm from the program can correctly tell us that number of mussels. Maybe it will still be good at predicting if that number of deep mussels is constantly. So what about them? They're alive. Are they active? No idea. No idea. And, what about the plants? What are they doing? So when we're thinking of different things that have happened, you guys have gotten quagga mussels in your lake, relatively recently, within the past 10 years. Also, your algal bloom problems have increased, just recently as well. How much of that has to do with you have a lot of quagga mussels, and maybe they're contributing to the problem. Maybe they can help stimulate release of the legacy phosphorus, so that stuff that people added to the lake before they knew that phosphorus was a problem. Maybe they're changing the nitrogen to phosphorus ratios by doing. I have no idea. all these things that everyone talked about could be influenced by them, but it's sort of cool, right? We might be able to solve the problem if we can figure out what they're doing. Maybe that will be something we can figure out. So I think, you know, the new normal, does it have to be this? You know, does it have to be something that we thought was just, you know, our fathers or grandfathers' phosphorus issue where we could reduce phosphorus and have the lake be clear instead of green? Like, people have done that and it worked, and now it's coming back. You know, some of it's maybe global climate change where we have less ice on, and then we have warmer water in the summer. That's causing diatoms and other really nutritious phytoplankton to lose competition and cyanobacteria to win. Could be mussels, could be nutrient input changes in, you know, types of nitrogen. But I think what it points to is that there probably are a lot of causes of what's happening with these harmful algal blooms, and unfortunately we have all these perturbations that, and we also have like stratification and all these upwellings of legacy phosphorus that occur. We maybe need to think we're not going to have one solution to how we fix the harmful algal blooms. Maybe it's like some kind of matching of different scenarios as to what, say, the 2 or 3 bigger contributors are in a lake, and how do we fix those? Can we prevent, say, the plankton from coming down and exploiting that quaggas are releasing all these nutrients by, you know, having in shallow areas or in other areas some mixing and so forth? Who knows? But I think what this was a good example of is sometimes you do something that you think will be simple, just parameterize the model, it can do that, but we also may have found some other things that are contributing to the problem, maybe helping to release or hurting us by releasing some of the nutrients in the sediments. We don't know. I think that's going to require some in situ, like in the lake work, to see what's coming off the bottom of the lake and what nutrients are being released. Some experiments. I've got actually later today I'm supposed to go out to Oneida Lake and help a student collect some mussels for an experiment. We're gonna, like, you ever heard of an ant farm when you were a kid? So we're gonna build like some quagga mussel ant farms, and we're gonna see, like, if we put them at the top in different densities, like, what do they do? So, and there's some students here who I know are like jazzed about thinking about that in the lake. So see if they can provide funding to do that. But we'll watch them and see if they move around and if they release phosphorus. So I just want to thank the whole suite of people. Matt Cohen, the graduate student, who was really our GPS whiz and so forth, GIS whiz, Bob Pedian and Alex Romer, the divers, and a lot of undergrad volunteers who are great. We're really lucky to have such great undergrads at ESF who want to make the world a better place and work really hard and don't care if they're mucky and sticking their arms up to hear the sediments. So I also told Nancy that I would plug the Diet for a Small Lake book. It's a really, really approachable, sort of limbological book. If you guys haven't seen it, you can actually get it for free online. Scott Kishko, he was the first author of 100 people. BC has a place contributed. And you can also buy it off the nice Nice site, listed there for $10 for first copy. So that's what I've got, and thanks for listening.
I have a couple of quick questions. How do you propose a way to determine how deep the mussels extend into the deep sediments?
Yeah, well, it's deeper than my arm. So
you think coring is an
I
option?
think coring is probably up. Yeah.
Okay. You have one, Jim?
I did have one, but I think she answered it. Since quagga mussels have recently appeared along with HABs, are quaggas the new livestock which are releasing legacy phosphorus which contribute to HABs?
No, but it's possible, and it's possible especially if they're like helping, they're being like the earthworms of our lake maybe, which is good if it's your garden that they're releasing all this, but it might not be good if it's the lake.
Thank you. Very good information. Thank you all for hanging in there.